us today. It is a true pleasure and honor for us to be here with you today to tell you a little bit about the work that we're doing to improve the care of patients with gastrointestinal cancers. It's a true honor to be here as part of the new members of the FEELUX team, and I'm excited to tell you about where we're going and what we're going to be doing. So first, let me introduce myself. So I am the professor and chair of the Department of Pharmacology and Experimental Therapeutics at Thomas Jefferson University, and I'm the director of gastrointestinal malignancies for the Sidney Kimmel Cancer Center of Thomas Jefferson University, and I'm also the chair of the FDA Advisory Committee on Clinical Pharmacology and Pharmaceutical Sciences. I got my PhD at Thomas Jefferson University in 1980. I did postdoctoral fellowship work for the next 10 years with the Nobel Prize winner Fred Murad, who won the Nobel Prize in 1998. And then I went on to do clinical training as a physician for 10 years at Stanford. And I completed training in 1990 and then returned to Philadelphia to join the faculty of Thomas Jefferson University. It's been an exciting time since then in the development, the invention and development of technology that I will speak to you about today. As Mr. Ahn alluded, in fact, we have formed a company, Coagentus, with ourselves and our Korean colleagues. And Coagentus was formed to develop the technology, the vaccine technology, and the adoptive cell therapies that we're going to use for metastasis in gastrointestinal malignancies. Coagentus, as Mr. Ahn told you, is now the second largest shareholder of FEELUX. The R&D team that you see here is formed by myself and my colleague, Adam Snook. Dr. Snook and I run a research enterprise of 20 investigators at Jefferson, and there's another 20 to 30 individuals, scientists and physicians who work with us to institute and conduct the clinical trials that form the core of clinical development of the vaccine and adoptive T-cell therapies that I'm going to talk to you about today. And on the right-hand side of the slide, our main scientific areas is beyond vaccines and adoptive cell therapies to treat metastasis in GI cancer. We also work on obesity and metabolic disease, neurological diseases, and inflammation. And I'm happy to talk to anybody about any of those areas that we work on later. So, for the portfolio of immuno-oncology on which we're working with FEELUX, the target population that you see up in the upper left-hand side, the target population are patients who are at risk or who have metastases from cancers of the colon, rectum, pancreas, stomach, and esophagus. We have created a portfolio of approaches that are cutting edge and on the advancing edge of the wave in terms of technology. We have more than 100 patents either issued or applied for in the overarching area of GCC biology and its application to cancer. We have more than 30 patents or patent applications for the applications that we're going to talk about today. And the two research fields that I'm going to describe to you today, one has to do with vaccines to prevent metastases in GI malignancies, and the other is adoptive cell therapies for GI malignancies. So for the vaccines, we are interested in taking patients who have been treated and are only at risk for developing metastatic disease and dying of their disease. We're interested in taking those patients and vaccinating them to prevent the development of metastasis and recurrent disease. For the adoptive cell therapies, we're using that to actually treat patients with established bulky metastasis, with the idea that we will be able to use those engineered cells to eliminate those metastases and potentially cure patients with metastatic GI cancer. So these are the technologies that we're working on. Currently, the most advanced of our technologies is the GCC vaccine that you see here, Ad535 HGCC Cadre vaccine. So we have done the preclinical, we've completed the preclinical work on this. We have brought, and I'll share with you some of that work. We brought that information to the FDA. The FDA granted us an IND, the ability to take that into phase 1, and we brought the first generation vaccine into a phase 1 trial of colon cancer patients. And what we learned in that trial was that the vaccine was safe and well tolerated, and in fact had immunological efficacy. It created immune responses to GCC as we hypothesized. And that technology is now positioned to enter into phase 2 this year, actually. We have manufacturing of drug currently ongoing, and I'll talk to you a little bit more about that in subsequent slides. The next technology that we are developing is the GCC CAR T-cell technology, or GCC adoptive cell technology, and here we're taking T-cells in this case from patients with metastatic disease. We're genetically engineering them to recognize GCC on their tumors, expanding those cells outside the patient's body and then reinfusing them so that those engineered T-cells will eradicate metastatic disease in these patients. And we've completed our preclinical studies with that and published those data and are positioned to approach the FDA to develop an IND and a phase 1 trial. And we're looking to start those trials in 2019. We have other applications that are in the pipeline. G-Biotics is focused on primarily intestinal health. I'm not going to talk a lot about that today, but those are technologies that are in the pipeline as part of the FEELUX portfolio. Just to give you a sense of the market and the value proposition that we're addressing here. So colorectal cancer, these are United States numbers. Colorectal cancer is the 4th leading cause of cancer and the 2nd leading cause of cancer death in the United States. 150,000 new cases every year, and about 50,000 of those patients die from metastatic disease. You can see pancreatic cancer. There's about 40,000 cases in the United States every year, and there's about 35,000 deaths from pancreatic cancer every year, so uniformly fatal. Gastric cancer, about 50,000 new cases each year in the U.S., and about 60% of those patients will develop metastatic disease and die. And then esophageal cancer, 15,000 new cases a year in the U.S. And again, uniformly fatal, about 15,000 deaths every year from esophageal cancer. And the take-home message here is that, or the important message here is that these diseases, particularly when they develop metastatic disease, are uniformly fatal, and there's no good treatment for any of these patients when they develop metastatic disease. And so there's an unmet medical need to develop new technologies to save these patients' lives and allow them to either be cured or manage their diseases, chronic diseases. And just to give you a sample of the markets that we're talking about, at the bottom here, you can see that the reimbursed price for cell therapies in the U.S. currently is roughly about $500,000 per patient. For vaccine products, it's about $100,000 per patient. So the populations, certainly in the U.S., are robust, and the markets are robust. So the work started with colorectal cancer, and as I've already told you, colorectal cancer is, in the U.S., the fourth leading cause of cancer, but the second leading cause of cancer death, again, in the United States. And about half of the patients that get colorectal cancer get metastases and they die of their disease. So we're looking to either prevent metastatic disease in these patients or treat metastases to cure the patients once they've developed metastatic disease. Some high-level highlights of the technology. So GCC, and you'll hear me refer to GCC a lot in this talk, GCC is the target for the technology that we work on. It's the target for the vaccine, and it's the target for the adoptive cell therapy. And GCC is a unique target because it's made normally in intestinal cells. I'll show you that in a minute. But because of its biology, it's unique in that it's also made almost universally by all primary and metastatic colorectal tumors. And because of that, we can use it to target drugs or cells or vaccines to attack and eradicate metastatic disease in these patients. So as I said, GCC here has been validated as an immunological target to prevent metastases in this disease. And Viral Gene has sublicensed that technology, and that's now a technology that is in the FEELUX portfolio that we're developing. As I indicated, all the preclinical work has been done. This has been through phase 1 study. The FDA has reviewed it. We've had our end of phase 1 meeting, and we are now preparing for our phase 2 trial to initiate in 2018. That's to prevent metastatic disease in patients with GI cancer. The next technology on the drawing board here is the GCC CAR T-cell technology. This technology has been through preclinical studies, very successful. I'll show you some of the data from preclinical studies. We're going to use that data to go to the FDA and achieve an IND to start a phase 1 trial in 2019. So what is GCC? So GCC is, this is very thin. So GCC, you can see it, that's an intestinal cell. The pipe of the intestine is at the top of the slide. The wall of the intestine is at the bottom of the slide. You can see that GCC is made in the tops of these intestinal cells. So it's made in the intestine, and that biology that it's made in the intestine gives GCC special characteristics that allow us to target it in metastatic disease, eradicate or kill tumor cells without harming the intestine. One of the characteristics is that it has a selective tissue expression. Primarily, GCC is made in intestinal cells from the top of the intestine all the way down to the distal rectum. All the cells in the intestine make GCC. And it's also made by metastatic, primary and metastatic GI, colorectal cancer cells, and other GI cancer cells. So it's tissue specific, it's compartmentalized. This is an important concept to understand the biology of GCC and why it's a good target. It is made inside the intestine, and the intestine is an immunologically privileged site. It has its own immune system that's separate from the rest of the immune system on the inside of the body, and so GCC is typically not recognized by the inside immune system inside the body. It's only expressed on the inside of the intestine, and that allows us to capitalize on its not being made normally on the inside of the body and making immune responses that kill cancer cells. I'll talk a little bit more about that in a minute. So compartmentalization And then it's universally overexpressed in GI cancers like colorectal cancer, pancreatic cancer, esophageal cancer, and gastric cancer. You can see there on the far right the intense brown staining. That is a malignant intestinal gland. That's GCC staining in the primary tumor that you see there. That's true in both universally, 100% of the time. Primary and metastatic colorectal cancer cells overexpress GCC, making it a great target. So, as you know, immunology has transformed the management of cancers, and you hear about it in the press continuously. It really has revolutionized the management of patients with cancer. You can see it was the breakthrough of the year in 2013. And this is a complicated slide that I'm not going to describe to you, just to tell you that these are the sequence of cellular events that occur in the activation of the immune system to attack a cancer cell. Figured out how to use GCC biology and GCC technology to harness the immune system to either prevent or kill metastatic disease arising in the GI tract. And you can see here there are 3 applications that we've developed. One of them is a vaccine for the secondary prevention of metastases. One of them is adoptive cell therapy to attack established metastases in patients. And then the other is an antibody-drug conjugate technology. Antibody-drug conjugates are sub-licensed to Millennium Takeda Corporation, excuse me, and they have this technology now. They have advanced it into phase 1 trials, primarily in the U.S. So they have these trials ongoing right now, which is interestingly a validation of the approaches that we're taking using GCC as a target. So let me tell you a little bit about the first 2, vaccines and CAR T cells. So this gets back to the concept of immunological compartmentalization. GCC is made on the inside of the intestinal tract, and because of that localization, and it's not being made on the inside of the body, that conveys 2 advantages to GCC as a target. One advantage is because its expression is confined to the inside of the intestine, it's not recognized as self by the immune system on the inside of the body. So that's one important benefit, and we can capitalize on that and try and generate immune responses that will kill tumor cells on the inside of the body because tolerance barriers are low. And the second part is there are 2 immune systems, one for the inside of the intestine, one for the inside of the body. And these 2 immune systems don't talk to each other. There's no communication. So when we create an immune response on the inside of the body to GCC that fights and kills metastases, those immune responses will not cross over inside the intestine and kill the intestine. So it allows us to generate on-target, on-tumor effects without subsequent adverse events on target in the intestine. So this is some of our preclinical data with GCC technology, GCC vaccine. So this is our first generation GCC. This is actually the vaccine that went into patients in phase 1. And you can see here, look at the lungs. So the lungs on the bottom, the lungs on the right are from an animal that was immunized with GCC. Lungs on the left are from an animal that was immunized with control vaccine. You can see that the control vaccine animal had lots of tumors in their lungs, and the GCC vaccine mice have virtually no tumors in their lungs. In the upper right, you can see that animals that are treated with the GCC vaccine that have heavy metastatic loads in their lungs, they live longer, and about 30 or so % of them are cured of their metastases. And all of that happens, down in the far right, all of that happens without initiating inflammatory bowel disease or adverse events in the intestine. As I said, we use these data to present to the FDA the idea of bringing this vaccine into a phase 1 study in patients with colon cancer. We did that, and in fact, we were able to demonstrate that the vaccine is safe, it's well tolerated, patients don't develop inflammatory bowel disease when they're immunized with GCC, and the GCC vaccine promotes an immune response against GCC in patients. You can see that in the middle graph in the red. Those are patient responses to the GCC vaccine, to GCC in the vaccine. And so we demonstrated that it's safe, well tolerated, and immunologically effective. We're using these data now to move into a phase 2 trial. As I said, we have we are currently manufacturing the new vaccine to start those phase 2 trial, that phase 2 trial. This is just some information on the second generation vaccine that we're having manufactured. It is second generation because we've modified it. We're now using a slightly modified, genetically engineered virus that allows us, instead of treating 50% of the population, we'll be able to treat 100% of the population effectively with this new virus. So we've really expanded the population that we will have the ability to treat to prevent metastatic disease. And this is the vaccine that's being manufactured that we will use in the phase 2 trial. Moving on from the vaccine to CAR T cells, where the vaccine is designed to prevent metastatic disease in people who are at risk for developing metastases. CAR T cells, in contrast, are designed to treat patients who already have metastases and will die of their disease. These cells are used to treat those patients, to eliminate the metastases, and potentially cure those patients of their metastatic disease. So the CAR Ts that we're using are actually third generation in the cartoon there. It's the far right cartoon. And it's third generation because the structure of the chimeric antigen receptor has an antibody head on the outside of the cell, and it has 3 signaling domains on the inside of the cell that allows the cells to better respond, more effective at killing cancer cells, and they persist for a longer period of time. As I'll show you in a minute, those characteristics actually allow us to provide lifelong protection to the animals in our preclinical studies against metastatic disease. So the idea is that a patient has metastatic disease in their liver. You can see that here. In these patients, we will take their T cells out of their bodies and we will engineer them to express a chimeric antigen receptor, or a CAR, chimeric antigen receptor. And that chimeric antigen receptor will have the head of a monoclonal antibody that recognizes GCC in our application. We'll take these genetically engineered T cells and we'll expand them. We'll make millions of them outside the body of the patient. And then we'll take these engineered T cells that will recognize GCC. Each one of these is a smart bomb. They'll home in and find cells that are making GCC and essentially blow those cancer cells up. We'll put them back into the patient. They will circulate and find metastatic disease and kill the GI cancer metastases, leaving the normal liver, in this case, the normal tissue, the normal liver intact. And so with this approach, we are looking to cure patients of their metastatic disease. These, to summarize this, again, high-level results from our preclinical studies. We have created mouse T cells that have mouse CARs recognizing mouse GCC. They were highly effective at eradicating and curing animals of their bulky lung metastases from colorectal cancer. And we've made T cells that have human CARs and put them in mice that carry human colorectal cancer metastases, human colorectal GCC. And those CAR T cells are highly effective at eradicating bulky metastases in the lungs of these animals. And they do all of that without harming the intestines. So there are no adverse effects of these cells. So here's the data from the mice. Control is untreated. MS7 is a control CAR, not directed effectively to GCC. And the MS24 is a CAR T cell directed to GCC. You can see in panel A, the MS24 is very highly effective at eradicating metastases in the lungs of these animals. In panel C, you can see that CAR T cells, treatment of these mice with bulky metastases effectively cures 80 to 90% of the mice of their bulky metastases. And it does that without causing any adverse events. Moreover, importantly, these CAR T cells essentially protect the mouse for its entire life. We can take the animals that survive in panel C after getting CAR T cells out a couple of hundred days and then re-challenge them with metastatic colorectal cancer cells, and those animals effectively reject the new metastatic colorectal cancer cells. You can see that in panel D, with more than 70% of those animals cured of their disease for their entire lives. So we think this technology is a particularly promising technology to eradicate GI cancers that make GCC without harming normal tissues. And so this is the clinical development plan, high-level clinical development plan for the adoptive cell therapy. We're in the final stages of identifying the optimum CAR structure that we want to go into patients with. You see here. We're then going to have these CAR constructs, so this is the drug. We're going to have these CAR constructs generated by a commercial vendor under GMP conditions. Once we get the GMP manufacturing of the CAR, and that takes about 6 to 9 months, once we get the genetic CAR construct in lentivirus ready for use in patients, we will take the information that we have from our preclinical models, take the manufacturing data for the lentivirus CAR T cell constructs, and we'll bring that to the FDA to get an IND to allow us to do our phase 1 trials. And our current concept of the phase 1 trial for our CAR T cells will be a basket trial in which we obtain T cells and genetically engineer them from patients with metastases from the colon, the rectum, pancreas, stomach, and esophagus to identify the best patient populations who are most sensitive to CAR T cells for eradication and elimination of their metastases. So I want to thank you again for your attention. I described a little bit to you about a very high-level description of our GCC portfolio of technologies for use in patients who are at risk or have metastases from GI cancers like colon and rectum, pancreas, esophagus, and stomach. We are looking forward to working closely with FEELUX on the clinical development plans for these technologies, and I will certainly stop here and turn the podium back over to Mr. An. Thank you very much.
Can CAR T cell kills other types cancer cells? For example, signet ring cell or well-differentiated ADLCA?
Yeah, so that's a really good question. The biggest successes so far for CAR T have been for liquid tumors, leukemias and lymphomas. And that's because they have very specific targets that you can hit with a CAR T cell without causing normal tissue damage. The limitation so far for solid cancers like prostate, breast, even GI malignancies has been to identify a good target that will allow you to kill only tumor cells and not normal tissue for these targets that are shared in normal tissue and tumors. The biggest limitation right now for CAR T, any adoptive T cell, in CAR NK cells, CAR T cells, any of these adoptive cell therapies for solid tumors has been the identification of the right target. We think, we didn't invent CAR, we invented GCC. It's the GCC that gives us the advantage to be able to go after GI cancers without causing normal tissue injury. And so, right now, our CAR approach is going to be useful for tumors that make GCC, so colon and rectum and esophagus and stomach and pancreas. It will not be useful for tumors outside the GI tract that don't make GCC. Those tumors will need to identify new targets to make new CARs to go after.
Thanks for your answer.
I can talk forever about that.
또 질문이 있으신가요?
제가 이제 조금 늦게 와서 이게 중복된 질문일 수도 있는데, 이거는 카티, 이거는 이제 전임상 단계고, 바이럴 진행하는 거는 이제 이상을 이제 디자인해서 이상 이제 곧 들어가면은 주가에는 일단 전임상 단계에서는 밸류보다는 바이럴 진을 통해 갖고 이상이 되는 게 이제 좀 더 중요할 것 같은데, 그러면 이제 스캇 박사님 그 연구팀이 두 개를 동시에 이제 진행이 되는 건지, 아니면은 어떻게 그게 가능하고, 아니면은 이게 바이럴진 이상을 하는 데 있어서 좀 그런 뭐 부분이 있는지, 이게 동시에 진행이 잘 될 수가 있는지, 이런 점이 궁금합니다.
주가에 대해서는 사실 뭐 회사에서 뭐 컨트롤하는 그런 분이 아니기 때문에 떠나서 또 질문하신 취지가 여기 두 가지를 동시에 진행할 수 있느냐, 여기가 질문하신 것 같고요. 그 부분은 저기 통역하시는 분께서 스캇 박사님께 좀 질문을 드리고 답변을 주시면 어떠십니까?
So these two clinical trials are actually quite different, and they ask different questions, and we are fully prepared to simultaneously have both clinical trials going. So we are currently engaged, as I said, in manufacturing the drug for the vaccine phase 2 clinical trial. We fully intend to initiate that. We have an IND for it, FDA IND. We fully intend to initiate that trial in 2018, probably in the fourth quarter of 2018. As long as we are resourced appropriately, we are also prepared at the same time to begin generation of drug product for the CAR T cell phase 1 trial. We have the patients and the investigators, the doctors that take care of those patients lined up, and we're ready to go. The study teams are all set. We are simply waiting for the resources to be made available so that we can order the drug. So the answer is yes, we are prepared to have both of these trials going simultaneously. Keep in mind, I'm not the one, they're not my patients, and so to do these trials, we interact with an army of doctors and their patients spread out across the largest healthcare system in the Delaware Valley, which is where I live. And so we have many, many patients, many, many doctors to do these clinical trials, and it's fully conceivable to be able to do these things simultaneously.